Hi everybody, my name is Josh Jacobs. I'm at the Department of Biomedical Engineering at Columbia University. The title of my talk today is Single-neuron correlates of spatial memory and navigation. My goal is to tell you about some of my lab's work studying direct human brain recordings in order to explain how the brain represents individual memories related to space and how we navigate spatial environments. To orient you to some of my work, imagine a situation that happens in my house every week. This is a bagel store at my house called Absolute Bagels, where I go bagel shopping for my family all the time, especially Saturday mornings. And getting bagels at this bagel store is a real memory challenge for me every day in my house. This is my daughter Sylvia. Here, Sylvia and each of Sylvia, along with my other kid Luke, each have their very particular bagels that they each want, that they each find are very important for me to get. Sylvia has her poppy seed bagel on the left here. Luke, who has stuff written on his face, has his egg bagel that he likes, and that's me on the right. Before the pandemic, when I used to shave my face, and I like bagels with seeds on them. These are everything bagels with seeds that I like. And it's really important that every day when I get my bagels, I have to keep, my kids are very passionate about which kind of bagel they each want. And not only does Sylvia want poppy, and I want everything, and Luke wants egg, but they each want their bagels at different places in their house, and they care about what kind of topping is on it, cream cheese or butter or nothing at all. So every week in my house, I have to keep track of how the bagels are thrown about my house and what the details are for each individual bagel order that I get. My kids are very passionate about this. So I get home, and Sylvia might ask me, where is the poppy bagel? Where is the bagel that she wanted? Where did it go throughout the house? And I have to keep track of, and this is an example of the kind of complicated memory tasks that the human brain is capable of. Because we not only have to keep track of where's Sylvia's bagel, where's the poppy bagel, but she can ask me lots of other questions. Where's yesterday's bagel? So that would be a bagel at a different moment in time. Which bagel is by the sofa? Where is the bagel kept throughout your house? When did I last eat on the sofa? And this silly little example about the kids' bagels in my house illustrate that some of the key aspects of memory, and in particular episodic memory, are the what and the when and the where information about memory. So the research in my lab, we study direct brain recordings from human neurosurgical patients, and what we're trying to do is understand how the brain represents individual memories and how we can use this information to, because we're hoping that if we can explain information like where our memories, how the brain represents memories for certain spatial locations, and how the brain represents information for certain kinds of bagels, like in this example, we can use this to explain, to help treat neurological diseases and to help people who have memory disorders. So the way we do this in my lab is with direct brain recordings from human surgical patients. These are epilepsy patients who have very severe epilepsy, and you can see an example of one of these people, their brains on the left-hand side here. Mild cases of epilepsy don't have electrodes implanted in their brains, but the patients with very severe epilepsy have electrodes implanted for about 1 to 3 weeks for seizure mapping. You can see an example of one of these patients in the middle panel of this plot here. Patients are implanted with 2 different kinds of electrodes, microwires, which record individual neurons, and that's what I'm going to focus on today. And other patients are also recorded with macro electrodes that record like an EEG signal, kind of like brain oscillations. And we have the patients in my lab do different kinds of behavioral tasks. And the kinds of behavioral tasks that they do in these studies are virtual reality video games, and you can see an example of one of these on the right-hand side. And we have them do these tasks on a laptop computer that we bring to their bedside, like you can see in the middle task there. We synchronize that laptop computer with the patient's recording system, and we get really wonderful and detailed data on how the brain operates with these more complex behavioral tasks that let us understand the neural basis of spatial memory. This is one of the tasks that I'm going to tell you about today. This is a task called Treasure Hunt. Treasure Hunt is a gamified spatial memory task. Each trial of the task involves patients driving around a maze, and they see a series of different treasure chests, and they have to remember which object is at which location in the environment. So now, as you can see from this trial, the subject is in a big, large, open environment. They drove to one treasure chest, and they're controlling their movement with a joystick. And they found a fan. It was at one location in space. They see a series of these across each trial. Then they are asked to recall where they saw each item. Here they see a memory task, and they're asked to respond to that task by moving this cursor with a joystick to indicate where in the environment the object is located. So they mark that location there with a circle, and then they provide feedback. And so you can see here the subjects saw two different locations in space. Remembered objects at two different locations in space. The fan on the right is marked with a green circle, which means that they responded pretty close to that, so they got positive feedback. The shield at the top, they responded not very close to that instead, so they lost 50 points, and you can see that from the red -50. And so by studying brain recordings from subjects doing these tasks, we get really wonderful data on how the brain can perform different kinds of behavioral tasks, and how the brain represents specific information to the neural representation of space during these tasks. So now I'm going to go through a series of different studies that my lab has carried out from subjects performing different versions of these kinds of tasks, and you can get a flavor for the kinds of data that we see in these recordings. To start with, here's an example of of two different neurons, what are called place cells and grid cells. Each of the, you're going to see throughout the talk, you're going to see a series of different overhead maps of different environments. The color so each square here on the left and on the right represents an overhead map of the environment. The color indicates the firing rate of the neuron while the person was located in that environment while they were navigating through space. So what you can see is that this background firing rate in blue indicates that this is one neuron in the hippocampus that we call a place cell, because this neuron is basically quiet. It has a low firing rate at many different locations in space, except for when they're in the middle of the environment, which is indicated by red here, where it has a high firing rate. So this is an example of a place cell, because this neuron is active at one place in the middle of the environment and basically is quiet when a person is at other locations. This neuron on the right is an example of what we call a grid cell. And these grid cells, maybe you've heard about them, but grid cells are neurons that are not just active at one location in space, but instead they are active at many different locations in space. As you can see around the arena here, there are these many firing fields where this cell is active, and it's called a grid cell because of these, because there are many different firing fields that are arranged in a grid-shaped pattern across the arena. So place and grid cells had been found before in humans, and my work, as you can see here in 2013, showed them for the first time in animals as well. Showed them for the first time in humans as well. Excuse me, all of my work is done in humans, but we try to build off of the work that's been done before in animals previously. And what you can see here is a taxonomy across different species of these different cell types. So how does the brain? So this is a taxonomy of the spatial representations that are used for animals during navigation, as well as we think during memory. So you can see animals have place cells and grid cells, and when it says animals here, that really primarily means rodent studies. We have place cells in rodents, grid cells. We have neurons that fire from particular directions. We have remote place cells, and we have oscillations as well, these large-scale network oscillations across the hippocampus. Humans also have such patterns, and there are two categories of patterns that have been found in humans before. In this second row, you have the human patterns, like some of those that I'm going to talk about, that represent patterns that are similar to what have been seen before in rodents. And then on the bottom row, there's a set of studies that I'm most excited about that show characteristics of the human brain during spatial navigation and spatial memory processing that are really specific to humans that have not been seen before in animals. So I'm going to go through a series of these studies overall that summarize each of these categories of findings from my lab. Okay, so the first study that I want to tell you about is some work that came out a couple years ago from my then graduate student Salman, and the question that this study examined is whether, so we know that humans have place cells that represent particular locations in space, and the question then is whether this so-called cognitive map of the environment is flexible according to what a person is doing. And what they're focused on. So we had subjects do a task where they're navigating through an environment, but on each trial they're trying to find a different object. So they are 4 different trials where they learn. There are 4 different kinds of trials. on each trial, they are each tasked with retrieving the location of one particular object. So here is the learning phase of the task, where the subject drives through an environment and they have to learn which object is located at which location in space. You hopefully saw at the beginning of there that there was a desk located at the beginning of the environment. And then during retrieval, on each trial, they are told what object they care about, but the object remains invisible, and they have to press a button when they're at that location. So there are 4 different hidden object locations, and they have to keep track of which object is at which location in space. So there are really 4 different conditions where we hypothesize that subjects might have different kinds of maps of the representations of the environment as they're moving. So you can see throughout the movement on this trial here, the target object was invisible the entire time. Okay, so the question then is, what do the neural data do when the subjects are performing this task? And in particular, were there some neural signals that differentiate based on what a person is trying to do in the environment? Okay, so this is a plot of a place cell that is active. And so on each, I'm going to show you a series of these plots, and on each plot, the horizontal axis is representing the position of the person in the environment. The vertical axis is the firing rate of one such neuron, and you can see that this neuron, as the person moves across space, has a high firing rate towards the end of the environment, the end of the linear track, and that's called the place field of that cell. Okay. So this plot here came from the set of trials where the person was looking for object number 1, as you can see over here. And when we split up the data and plot this result for each of the different object retrieval conditions, objects 1, 2, 3, and 4, you can see that this neuron has the same firing field based on irrespective of which object the person is looking for. So this is an example of a classic place cell where the neuron fires at the same location across all conditions. We also found these neurons, called memory trace cells, in the entorhinal cortex, which shifted the locations of their firing field based on what a person is trying to find. So this cell, you see that it has this peak here, and this particular neuron changed the location of its firing field so that it fired at the location that the person was trying to find in the environment. So here it shifted between object 1 and 2 and 3, and then it had a low firing rate during object 4, so it didn't select for object 4 in any way. And this is a common pattern. So a lot of these neurons, these are especially found in the entorhinal cortex, these so-called memory trace cells change their firing field based on what a person is trying to find. So what this is evidence is that the human cognitive map is not a static map that is only about encoding location in an environment, like what has been hypothesized based on animal studies, but instead the human neural representations of space is more flexible, and it can vary its firing depending on what a person is trying to do in an environment. So I think that's exciting because it's something new that's not been seen before in animal studies. Okay, another factor that another characteristic that a person needs when they're navigating throughout an environment is a set of neurons that encode where a person is pointed through space, where their sense of direction, if you will. There are 2 kinds of sense of directions that have been hypothesized based on a lot of studies in psychology and other sorts of studies. There are so-called allocentric directions, which is the coding of direction in terms of world-centered coordinates, such as north, south, east, or west. There are also other coordinate systems, such as so-called egocentric coordinates, which is not based on north, south, east, or west, like a compass, but instead is based on the direction relative to the heading at which I'm facing in. For example, if I have a memory of a bicycle being straight ahead of me and a bucket of water being to my right, those are so-called egocentric coordinates. So a question then is, how does the brain represent these egocentric coordinates? Because we know, as you can see from this paper, that both allocentric and egocentric information are relevant for both memory and spatial navigation more generally. Okay. Well, in this study, which was published last year with a postdoc in my lab, Lucas Kuhns, we found that humans have what are called egocentric anchor cells. That's what we call them. So an anchor cell is a neuron that, if I'm located at this white circle in the environment and I'm pointed in the direction of the blue arrows, an anchor cell activates according to the relative location in egocentric coordinates of landmarks relative to the position where I'm pointing. So if I'm facing in the blue direction here, and I have an anchor cell that activates when the gray landmark is on my left-hand side, then this neuron would have a high firing rate over here when that gray landmark is to my left-hand side, as you can see here. So each anchor cell is defined in terms of where what the landmark is that I care about and what the egocentric orientation is to that position. And this is very different from neurons that might be encoding north, south, east, or west, because those north, south, east, or west cells have been seen before in animals. So one such egocentric anchor cell is active when this location here in the blue circle is, and then if you look at the direction tuning curve, if it's located to my left. So that you're going to see a series of plots like this this rainbow plot on the right-hand side here, where A stands for ahead, B stands for behind, L stands for left, and R stands for right. And you can see the biggest bars in this plot are to the left-hand side, indicating that this is an egocentric anchor cell that that has a firing field for for a particular landmark being on my left-hand side, and that's an egocentric coordinate framework. We see a number of these representations across the brain. Here's another example of a neuron. So the left-hand plot is an overhead map of the environment. The anchor field, or the landmark that the cell cares about, is in the bottom right-hand corner of that environment, as you can see from the colored dots. And this neuron has the highest firing rate when this location, when this landmark is ahead of me and to the right, as you can see here. And this plot here is just showing the preferred directions of that, the preferred location, the firing direction of that cell when a person is at different locations across space. And here on the bottom is another example anchor cell, and this is a cell that's kind of interesting. I think this cell cares about a firing field that's in the center of the environment, as you can see from that black circle in the bottom left-hand corner, and it is active when this cell is behind me. So this is a neuron that is active when the center of the environment is behind me, when you're driving away from the center of the environment while you're navigating. These cells are prominent in the parahippocampal cortex, where about 20% of the cells are anchor cells. So this is a very robust phenomenon that we see over and over in this data set. Okay, in the last section of my talk, I want to tell you about a phenomenon of hippocampal theta oscillations. And this relates to a question of, so while humans or animals are navigating and doing spatial memory tasks, basically any task while the hippocampus is active, we often see this 8 hertz, this large-scale oscillation across the hippocampus. And it shows up at 8 hertz in rodents and maybe at slower frequencies in humans. And this theta oscillation is linked to a broad range of different behavioral processes, from navigation to memory, based on animal studies. And in this study, what we tested the hypothesis of is whether theta oscillations were involved for individual neurons to encode information about where a person was located. There's been, as some of you may know, in addition to the large body of work on how neurons encode information based on increasing the rate of their firing, there's a complementary set of work on how neurons code information via changing the timing of their spiking. So what we wanted to examine in this study is whether individual neurons might represent information about where a person is located in an environment by changing the timing of their of their action potentials, of their spikes. So we tested for the phenomenon. So there's a phenomenon called phase precession that has been seen before in a large number of animal studies. And here, in these two figures here, you can see examples of phase precession recorded in humans for the first time. Each row represents one neuron. And what we did is the colors plotting the firing field of the cell, the firing rate of the cell as the person traversed different locations in this environment. And you can see that there's this one firing field as indicated by the white circle in the bottom left-hand corner of the environment. And we plotted and in the middle panel, we've plotted not the rate of the cell's firing, but the phase of this cell's firing relative to ongoing oscillations in the theta bands. And on the vertical axis is representing the phase of the cells spiking, and the horizontal axis is representing the position of the person throughout the environment. And what you can see is that there are these diagonal clouds of the points. So as the person is going throughout each place field from the left-hand side to the right-hand side, you can see that the individual neurons are shifting. They're progressively shifting the firing, the timing, the phase of their spiking relative to the ongoing oscillations from later phases towards the top to earlier phases at the bottom of the curve. And that's the phenomenon what's called phase precession. And you can see that on the left-hand, on the top row and on the bottom row. So there are two different examples of neurons that exhibit this phase precession by changing the firing field of their spiking as a function of where the person is going in the environment. So this is an example in humans of phase precession, of phase coding for a person's spatial location, and this is a new kind of phenomenon that seems to go beyond what's been seen before from rate coding. And I think this is really interesting because this is like kind of a complementary way that neurons can encode information in the human memory system. We know that neurons can encode information by changing the rate of their spiking, and now we know also that they can change the timing, the phase of their spiking relative to these network oscillations, and that's a second way that neurons can encode where a person is located in the space. Okay. We also see something that we call phase precession for goal states. I'm not going to be able to go through this in detail now because I'm over time, I'm sure. But basically, we see neurons that fire that when a person is going to different goals in the environment, we see many neurons that change the phase of their firing also, depending on where in the environment a person was going to, goal 1, 2, 3, 4, 5, or 6. So again, this goal phase precession is another way in which neurons shows that phase precession is more relevant than just encoding locations, but it seems to be that phase coding in the human brain is a broader phenomenon that extends more deeply beyond just rate coding. Okay, so to wrap up, and anyway, to learn more about phase precession, you can take a look at our paper. It was published last year, and Qasim et al. Is the first author. You can take a look at that if you're interested. So I hope I showed you that humans not only replicate a lot of the results that have been seen before from the animal memory system, but we also seem to have some new kinds of remapping patterns that have new kinds of cellular firing patterns that have not been seen before from animal studies. We have neurons that remap across memory retrieval, as you can see here. The same I didn't have time to talk about it, but we've also seen that these same representations correlate with memory performance as well. We have these egocentric coding patterns by the Kunz et al. Paper, as you can see here. And then finally, humans show a lot of different patterns of theta oscillations across the human hippocampus. Not only do humans have these humans also have these fast and slow theta oscillations, which seems to be different from what's been seen before in animals. And we also show phase precession as well, as I mentioned on the previous slide. Okay, so that's all for my talk. Thank you for your patience. These are my clinical collaborators with which I'm very grateful for their help, and for the patients also for participating in this research with me. I'm grateful to the NIH for helping provide funding for this work, and the NSF and the Nurtec Brain Research Institute. This is my lab here. We do fun things, and you can see, if you're interested more in this research, you can check out our webpage, and you can follow me on Twitter as well. Thank you very much.